Research is the cornerstone of innovation in clinical practice, driving improvements in patient outcomes and shaping the future of medicine. Welcome to the Global Rhinology Network Topical Research Symposium. I'm Ashok Rokade from Winchester, UK, and it is my privilege to welcome you as a host. Joining me as co-hosts are Louise Melia from Bath, UK, and Professor Thibault Van Zele from Ghent, Belgium. This symposium showcases fascinating topics across 4 key modules: general rhinology, COVID session, and research in chronic rhinosinusitis. A special highlight is the PhD session featuring research conducted by enthusiastic young rhinologists who represent the future of our field. This symposium brings together leading global minds in rhinology who will highlight latest developments with a focus on translating research into impactful patient care. The Global Rhinology Network is dedicated to advancing knowledge innovation. Thank you for being part of this exciting journey. Thanks to Medtronic and Karl Storz for their generous support in making this symposium possible. Welcome to GRN Topical Research Symposium.
It's my pleasure to introduce Professor Laura van Gerven, also from Belgium, from University Hospital of Leuven. She is a staff member of the rhinology department over there and has an experience also in skull-base surgery. And she did a PhD on the mechanisms of allergic and non-allergic rhinitis. And she has done very interesting work in the previous years, especially on the visualizing how SARS-CoV-2 can attack the cells of the olfactory mucosa. So please, Laura, show us your latest results.
So good afternoon, everybody. Thank you to the organizers also for the nice invitation. And I had a great afternoon already. It was very interesting, all the previous talks. So thank you also to my co-presenters. So I'll tell you a bit what we did in the Anosmic 19 project. It's a project that we started in the beginning of the pandemic, and it stands for Analyzing Olfactory Dysfunction Mechanisms in COVID-19. As you can see on the bottom, it's teamwork. So I've not done all the work alone that I will present in the next 15 minutes. It is really a collaboration between, you can also see in the author list, a team in Frankfurt led by Peter Mombart, who is specialized in analyzing human olfactory mucosa, and senior scientist there, Mona Khan, and then people here in University Hospital of Leuven, led by myself, and also colleagues of the hospital in Bruges, also hospital in Belgium. So the results that I will show have been published last year in Cell, and the recent update 2 weeks ago in Neuron, actually. So I will guide you through the results and hope to convince you of what the virus is really doing at the level of the olfactory mucosa. So these are the teams that collaborated that I already mentioned. And Thibault Van Zele mentioned it in the introduction. So olfactory dysfunction in COVID-19 is something that we all know from our rhinology clinic. I don't need to go into detail about that. A little bit less in the Omicron era, let's say, but still we see a lot of patients that are still suffering from persistent anosmia or hyposmia or parosmia. And up until now, the mechanism of this olfactory dysfunction in COVID-19 is still obscure. Also, actually, for other types of post-viral olfactory dysfunction, we have very little knowledge, and this became very clear in this pandemic. Why is it actually that we have so little insight on the mechanisms of the olfactory system compared to the visual system or the auditive system? It is probably because that in humans, the olfactory system is difficult to study. If you want to take biopsies, for example, in living patients, you know where the olfactory cleft is, but it is very difficult. You cannot distinguish microscopically olfactory mucosa from respiratory mucosa. It's all pinkish there. And so actually the respiratory mucosa really embedded islands of olfactory mucosa. It's not one uniform sensory sheet like in mice, for example. So it is difficult to take biopsies in a guided way. It's actually a bit blind. And also in the olfactory mucosa, you have patches of aneuronal epithelium, so without olfactory sensory neurons, actually. Then the bulb. So the next step, actually, of course, in living patients, you cannot sample this structure because of the intracranial position. This would leave the patient with debilitating consequences, but you can do so in diseased patients. And so this brings us then to the post-mortem sampling. In this way, indeed, COVID provided us with a unique setting. We had a lot of patients who suffered from this and unfortunately also died of the disease. But an autopsy or a conventional autopsy obviously gives very long postmortem intervals because you have the transport to the morgue and all these kind of things. And also the tissue itself is prone to autolysis of cells, tissue. And if you want to study an RNA virus like the SARS-CoV-2, it is also something that is very sensitive to a long postmortem interval, of course. So that's why we got the idea to develop a workflow, actually, 24/7, that was initiated by a phone call from an ICU physician or a physician on the COVID ward, or for the control patients. We were contacted by the people of the transplant team, and they informed us. And when the family allowed us to go to the patient, we performed actually with a mobile tower, and the equipment was installed on this tower. We went bedside to the ward or to the ICU and performed actually an endoscopic endonasal transcribriform approach, like we do this in skull-base surgery, and took in this way samples from the respiratory mucosa, olfactory mucosa, and olfactory bulbs, and also adjacent brain. I have here a small video just to give you an impression of the procedure and what type of tissues we collected to investigate then later on a histological and molecular level. So you have a right nasal cavity here. You see the inferior turbinate, the middle turbinate, and the superior turbinate. The septum is, of course, on the right side, and the olfactory cleft is located medial from the turbinates, the middle and the superior one. We always started with a swab. We did sequencing on this, so to know the variant of concern and also to have an idea of the CT value, to know if the patient was still infectious, etc. This is then a complete resection of the inferior turbinate, and we preserved it actually in one block, put it in the paraformaldehyde for fixation, and only dissected later on after fixation the mucosa from the bone for the further analysis. We did the same with the middle and the superior turbinate. As you know, there might be some olfactory mucosa on the medial side of the superior and the middle turbinate. This has been reported. Then we had some more space also to harvest olfactory mucosa. So we really took a wide epileptical incision, actually covering the superior part of the septum, the whole cribriform plate, and then ended just medial from the attachment of the middle turbinate. You see that we dissect in a superosteal plane, and that you really see the fila olfactoria running through the cribriform. So we transected the fila and then removed actually in piecemeals the resection, the olfactory cleft mucosa. And then we did what we would normally do. We call it a FES procedure, but of course, just to expose the skull base. And we did a kind of adapted transcriptiform approach. We didn't need the entire opening. So here we resect the remaining attachment of the middle turbinate. And then we will make a small window just to expose the olfactory bulb. We used hammer and chisel to avoid actually a drill because this can generate aerosols. And as you know, those patients are still contagious, and we want to avoid that. So you see the dura mater here, the bone has been removed, and then we will incise the dura to really get full exposure of the olfactory bulb. You see here the posterior part. Once it's open, we will detach it from the surrounding tissue, so the brain, and then in an atraumatic way try to detach it as posterior as possible to have also part of the olfactory tract. We then also took samples of the brain, but for the sake of time, I will maybe stop here. You can also watch a movie online. What kind of patients did we include in the first cohort? So 70 COVID patients and 15 controls, 2 convalescent cases, and really 68 that died, that were hospitalized for their COVID infection. The people that were in the cohort were mainly men with a lot of comorbidities, as you can see, average age of 80, so old men. And you can see here that a significant amount of patients died within the first 14 days after the diagnosis, which was made with a PCR of a nasopharyngeal swab. We could reduce, by using this technique, the postmortem interval to 71 minutes, so more or less 1 hour, which is significantly lower than in a conventional autopsy setting. As already mentioned, we collaborated then for the further analysis with the Max Planck Research Unit for Neurogenetics in Frankfurt, and did there, or the H&E was done here in Leuven, but the immunohistochemistry and RNA scope analysis was done over there. We checked with the H&E for structural changes. We tried to identify different cell types and searched for a general inflammatory response in both respiratory and olfactory mucosa, but also olfactory bulb. Then, as already mentioned, for the in-depth analysis of the tissues, the next step was a combination of immunohistochemistry and RNA scope. RNA scope is actually as in fish, so single molecule in situ hybridization. And I will show you briefly why it is such a great technique to study a single-stranded RNA virus that we try to find back in those samples. So you really have a unique sequence of amino acids that you can target with this double Z probe actually. And after binding, you will have kind of a Christmas tree that binds on this double Z formation and will actually light up like a Christmas tree. And so the presence of this RNA will be represented by a punctum, an immunofluorescent punctum, or if there is more, several puncta. So just to give you then an idea how it looks like. So these were the cell types that we studied in the respiratory epithelium. So we studied the ciliated cells, the goblet cells, and the duct cells. And you can see here in Roman letters are the markers that we used for immunostaining, and in italics the ones for the RNA scope experiments. And this is how it looks on H&E. So you have the respiratory epithelium separated by the basal membrane, and then the subepithelial space or the lamina propria containing the ducts and the blood vessels. This is in a control sample, so no COVID. You have EpCAM as an epithelial marker actually, and the MUC5EC for the goblet cells. You can really see that the mucus is blue. And then in combination with the RNA scope, so the puncta, this is RNA scope for FOXJ1, which is a marker for ciliated cells, and you really see that they co-localize. This is really a way to identify the cell type and see the surrounding structures in great detail. So this is how it looks then in a COVID patient. This was case 68. So we have the FOXJ1, so the puncta RNA scope again for the ciliated cells. The black is, of course, the basal membrane. There are no nuclei there. And then we used nucleocapsid staining, immuno, for the virus. And you can see that we have some heavily infected epithelial ciliated cells. And if we add then RNA scope for the virus, and here what is great on this probe is that it is a sense probe. So actually you have SARS-CoV-2 is single-stranded RNA. During the replication phase, you have a negative strand, and actually it's this one that can be detected by the SARS-CoV-2 RNA scope probe for the sense. So it's really only present in active replicating cells. So it's not only infected the cell, but there is a viral replication. And you can see that the dots are perinuclear, which is very typical. This is another case, COVID-7, also respiratory mucosa. So again, a patch of more infected, or at least there are more infected ciliated cells. The previous one was a bit more patchy. And here you see that there is again a lot of active viral replication in this part of the respiratory mucosa. We used actually 7 probes to really have high sensitivity, specificity, and you can see that there is very little background. Then we moved on to the olfactory epithelium, because I told you already, it are really islands inside the respiratory mucosa to search for the target cell type in the olfactory epithelium. And there we used markers for sustentacular cells, which are here in purple, the supporting cells of the olfactory sensory neurons, which are here depicted in yellow. And again, so the Roman ones are the immunomarkers and the italic ones are the RNA scope probes. We used various RNA scope probes for different maturation status of the olfactory sensory neurons. We used also markers for the duct cells and cytokeratin 5 or 6 for basal cells. And some imagination, the sustentacular cells look more like a Prosecco glass, and the OSNs more like cherries. And this is important in the next slides. So here, just to orient yourself, an HNE, you see that the olfactory epithelium is a bit thinner. Also, the basal membrane is thinner. The morphology is a bit different, of course, but also then a big lamina propria below it with the ducts. So again, first a control to make you familiar with the stainings. So cytokeratin 8 will stain sustentacular cells or supporting cells. And then you have OMP, which is really a specific OSN marker. And you can see the cherries in between the sustentacular cells. So the sustentacular cells really enwrap the OSNs. So this is then a specific olfactory receptor gene that we used. You have 389 in humans, and this is one of them. This is then a long stretch of a COVID case, olfactory mucosa. You see a lot of olfactory receptors lighting up in green because there are so many dots or puncta actually. And we used a mixture actually of various olfactory receptor genes and cytokeratin 8 for the supporting cells. And then again, the virus replicating probe, RNA scope probe, is really abundantly present in a lot of cells. If we zoom in then, because we are interested, what is the target cell type specifically, then we see actually that the OSNs are never containing the red dots of the replicating virus, that it is always in the sustentacular cells that are just next to them. And here you can really see one sustentacular cell really positive for nucleocapsid. So the blue is the immuno, and then the RNA scope for the active viral replication. What did the results of the bulb sample show us? Actually, that you can see here an entire bulb and I will be very brief for the sake of time, actually we could never find any virus inside the bulb. So the neuronal tissue was completely spared in regard to the bulb. So in conclusion, we included 30 patients, but actually only 70 patients, but actually only 30 contained the virus still in the sample. So the others were too long after the diagnosis. We found it back until 16 days and viral replication only until 9 days. There is one exception, that's the patient here at the bottom, but this was a organ transplant recipient, so was under immunosuppressive medication and therefore probably containing the virus a lot longer. We had the non-variants of concern, so really in the beginning, the first and the second wave in Belgium, and also the alpha variant later on. In the new study, we have Delta and Omicron. This is the graphical abstract with the conclusion. So in the respiratory epithelium, we have infected ciliated cells, so that's the target cell type of the virus. In the olfactory epithelium, we have the sustentacular cells. And in the olfactory bulb, we don't see it inside the bulb, but in very selected cases, we could find the virus back on the leptomeningi covering the bulb. So we then try to find out if it can be a port of entry to the central nervous system. And this is what we just published 2 weeks ago. So I invite you to read this manuscript if you're interested in it. And then I want to thank the whole team, the Anosmic 19 team, with mainly Wout and Marnik, who collected with me the samples here in Leuven, and then the people in Frankfurt, Peter Mombaerts, Mona Kahn, our pathologists, and also people from ICU. It's been a team effort, and I want to thank all of them. And of course, also you for your interest in attending this session. Thank you very much.
Thank you, Laura, for this interesting talk, and also for the risk you took in the beginning of the pandemic by doing this type of surgery, which was not evident and without any risk at that moment. Are there questions from the people attending here? Do you think this effect is specific for SARS-CoV-2, or do you think that other viruses have the same, let's say, capacity?
So yeah, we will have to find out. Of course, we have ethical approval also for influenza cases, but there was no influenza the last winter here in Belgium. So maybe we will do some cases this winter, and then for sure regarding the barriers. So what we showed actually in the second manuscript, we try to identify really the protective layers that can stop the virus. And I think that is really something that is translatable probably to other viruses. So actually the brain is super well protected. Also in the olfactory tract, you have olfactory nerve fibroblasts that really protect the axon bundles before they run through the cribriform plate. And I think this is probably not specific for SARS-CoV-2, but also true for other viruses.
Okay, I see Sarah appearing.
Yeah, I was just wondering, what do you, because you don't see the virus in the olfactory bulb, so do you have any kind of idea of why you see this anosmia then occurring?
Yeah, so we think that, but of course it's not really, we don't elucidate the mechanism completely. But so the sustentacular cell is a cell that has been very poorly studied, but it feeds probably the OSNs. So it provides, for example, in rats at least, ATP that make the how do you say this, the microvilli move, etc. And probably if you have really a complete destruction of the surrounding OSNs, this might influence the function of the OSNs. And this is also probably because, yeah, the majority recovers, luckily, so why you see a good recovery rate. Once they are restored, they will support the OSNs again, and people will have recovery of their sense of smell.
And do you have any information of these patients on whether they were actually anosmic at the time of yeah?
Mm-hmm. Yeah, so this is indeed a major limitation that is a bit inherent to the fact that they are severely ill patients that eventually die. So for some, we have some data at the moment of admission, but so yeah, now we are entering a new phase here, and we are in the smell and taste clinic in this well-defined patients with olfactory dysfunction, trying to take biopsies to get insight and to have the combination of both.
So Laura, we have come across a different type of virus strain, which have a different severity clinically. So did you have any data relating to that?
Mm-hmm.
Did you have enough patients from different strains?
Mm-hmm. So we have now 45 patients with so 20 with Delta and 25 with Omicron, BA1 and BA2 included in the cohort. And actually, we still find it in the olfactory epithelium, also in the cases that we have, but we see a lot less infected sustentacular cells. So in the cases where we see it, we still see it in that area, but yeah, it's difficult to do quantitative studies on these samples actually. So I don't dare to say, yeah, a lot
so
about
prima facie,
it.
the patients were less affected. Did they have a less sustentacular cell involvement?
Yeah, it was very difficult to find in the cohort with Omicron patients infected sustentacular cells. But I mean, in general, it's difficult to find it because there is probably only a small time window. So I don't dare to say too much about this if it's really less, but probably it is. But I cannot say that it's really quantitatively proven from our samples, actually.
Okay. Thank you. That's really interesting. And the efforts you have taken and the risk yourself by obtaining these samples is really phenomenal. So thank you for presentation.